friends to a new session on genomics and proteomics. In this session, we will discuss gene isolation, isolation of genes from genomic DNA by cDNA and exon traps. Gene isolation. What are genes? Genes are the basic unit of a cell that passes on the traits of parents to the next generation. Genes are pieces of DNA, or genes are part of a DNA molecule which codes for one polypeptide. They have information for making specific proteins that control specific traits or activities. Examples of traits controlled by genes are eye color, food size, and height. Examples of activity include the growth and repair of cells. Genes are also called as alleles or traits. Genes are actually a small string of bases on a DNA chain. So, a DNA chain has many genes and these DNA are located in the chromosomes. Chromosomes are in the nucleus of all cells. A full understanding of a gene or the entire set of genes in a genome requires that they be isolated and then studied intensively. Once a gene is isolated, one can determine both its biochemical structures and its function in an organism. One of the goals of biochemistry and molecular genetics is to assign particular functions to individual or composite structures. With the development of rDNA or recombinant DNA technology or molecular cloning enable researchers to isolate any gene from any organism from which one could isolate intact DNA or RNA. The full potential to provide access to all genes of organisms is now being realized as full genomes are sequenced. Gene isolation or DNA isolation is needed for genetic analysis which is used for scientific purposes such as introduction of DNA into cells and animals or plants for diagnostic purposes like gene cloning. It is also used for pharmaceutical industry to identify point sources for hospital and community based outbreaks and to predict virulence of microorganisms. The most important application for the isolation of genes is in forensic science, which needs to recover DNA for identification of individuals, for example, rapists, petty thieves, accident or war victims, and paternity determination. Isolating the genetic material or DNA from cells, it may be bacterial, viral, plant or animal, involves three basic steps. The first one is rupturing of cell membrane to release the cellular components and DNA. The second one is separation of nucleic acids from other cellular components, and the final one is the purification of DNA. Isolation and purification of genomic DNA. Genomic DNA is found in the nucleus of all living cells with the structure of helical ribbon. The isolation of genomic DNA differs in animals and plant cells. DNA isolation from plant cells is difficult due to the presence of cell wall as compared to the animal cells. The amount and purity of extracted DNA depends on the nature of the cell. The method of isolation of genomic DNA from a bacterium comprises bacterial culture growth and harvest, cell wall rupture and cell extract preparation, DNA purification from the cell extract and concentration of DNA solution. Growth and harvest of bacterial culture. Bacterial cell culture is more convenient than any other microbe as it requires only liquid medium or broth containing essential nutrients at optimal concentrations for the growth and division of bacterial cells. The bacterial cells are usually grown on a complex medium like Luria bertani or LB or nutrient broth NB. Later, the cells are separated by centrifugation and cell extract is prepared by lysis of cell wall to release the genetic material. DNA can be achieved by physical method by mechanical forces, chemical method by metal chelating agents like EDTA and surfactant, that means the SDS or enzyme like lysozyme. Catalyzes the breakdown of cell wall, that means the peptidoglycan layer. EDTA or ethylene diamine tetraacetic acid is a chelating agent necessary for destabilizing the integrity of cell wall or inhibits the cellular enzymes that degrade DNA, and SDS or sodium dodecyl sulfide helps in removal of lipid molecules and denaturation of membrane proteins. Generally, a mixture of EDTA and lysozyme is used. Cell lysis is followed by centrifugation to pellet down the cell wall fractions, leaving a clear supernatant containing the cell extract. Purification of DNA. In addition to DNA, a cell extract contains significant quantities of protein and RNA, which can be further purified by extraction with organic solvents and enzymatic digestion for the removal of contaminants. Extraction of organic solvents involves the addition of a mixture of phenol and chloroform in the ratio 1:1 to the cell lysate for protein separation. The proteins aggregate as a white mass in between the aqueous space containing DNA and RNA and the organic layer. Repeated phenol extraction is not desirable as it damages the DNA. Treatment of lysate with protease in addition to phenol or chloroform ensures complete removal of proteins from the extract. The RNA can be effectively removed by using ribonuclease, an enzyme which rapidly degrades RNA into its ribonucleotide subunits. DNA is collected from the upper aqueous phase by alcohol, that means the 2-propanol or ethanol precipitation, followed by rehydration. After air drying for 5 minutes, the DNA was resuspended in 30 microliter of TE buffer. The isolated DNA was confirmed using agarose gel electrophoresis. Agarose gel electrophoresis. Agarose gel electrophoresis is a method for separating and visualizing DNA fragments. The fragments are separated by charge and size and move through agarose gel matrix when subjected to an electric field. The electric field is generated by applying potential across an electrolyte solution or buffer. When boiled in an aqueous buffer, agar dissolves and upon cooling solidifies to a gel. For the visualization of genomic DNA bands, 1% agarose gel is used. 1% agarose gel is prepared in 1x TE buffer which have a pH of 8. 6 microliter of 10 milligram per ml of ethidium bromide or ETBr is added to the gel before casting. It is used because upon binding of the molecule to the DNA and illumination with a UV light source, the DNA banding pattern can be visualized. 1x TBE buffer of pH 8.2 is used as electrophoresis buffer. After placing the gel in electrophoresis chamber filled with electrophoresis buffer, load the samples, that means the 10 microliter DNA in addition with 10 microliter loading dye, and carry electrophoresis at 50 volt for 30 minutes. The genomic DNA bands are visualized using the gel documentation system. The next is isolation and purification of RNA. RNA or ribonucleic acid is a polymeric substance consisting of a long single-stranded chain of phosphate and ribose units with the nitrogen bases adenine, guanine, cytosine and uracil bonded to the ribose sugar present in living cells and many viruses. The steps for the preparation of RNA involve homogenization, phase separation, RNA precipitation, washing and redissolving RNA. The method for isolation and purification of RNA are as follows. The first one is organic extraction method. Second is filter based spin basket formats. Third is magnetic particle methods. And the fourth one is direct lysis method. Organic extraction method. Organic extraction method involves phase separation by addition and centrifugation of a mixture of a solution containing phenol and chloroform in 1:1 ratio and aqueous sample. Addition of chloroform forms a colorless upper aqueous phase containing RNA, an interface containing DNA and lower phenol chloroform phase containing protein. RNA is collected from the upper aqueous phase by alcohol, that means the 2-propanol or ethanol precipitation followed by rehydration. One of the advantages of this method is the stabilization of RNA and rapid denaturation of nucleases. The next is direct lysis methods. This method involves use of lysis buffer under specified conditions for the disruption of sample and stabilization of nucleic acids. If desired, samples can also be purified from stabilized lysates. This method eliminates the need of binding and elution from solid surfaces and thus avoids bias and recovery efficiency effects. Methods for isolation of gene or DNA fragment of interest. Every gene manipulation procedure requires genetic material like DNA and RNA. Nucleic acids occur naturally in association with proteins and lipoprotein organelles. The dissociation of a nucleoprotein into nucleic acid and protein moieties and their subsequent separation are the essential steps in the isolation of all species of nucleic acids. Isolation of nucleic acids is followed by quantitation of nucleic acids, generally done by either spectrophotometric or by using fluorescent dyes to determine the average concentrations and purity of DNA present in a mixture. Isolating the DNA from cells involves the rupturing of cell membrane to release the cellular components and DNA, separation of the nucleic acids from other cellular components and purification of nucleic acids. Genomic DNA is found in the nucleus of all living cells with a structure of double-stranded helical ribbon. The isolation of genomic DNA differs in animals and plant cells. As compared to animal cells, DNA isolation from plant cells is difficult due to the presence of cell wall. The amount and purity of extracted DNA depends on the nature of the cell. The method of isolation of genomic DNA from a bacterium comprises bacterial culture growth and harvest, cell wall rupture and cell extract preparation, DNA purification from the cell extract and concentration of DNA solution. Gene isolation based on protein sequence. One of the more elaborate examples of inverse genetics is the situation where the protein of interest has been purified, but the gene that encodes that protein has not been identified. Protein sequencing is a technique to determine the amino acid sequence of a protein as well as which conformation the protein adopts and the extent to which it is complexed with any non-peptide molecules. The two major direct methods of protein sequencing are mass spectrometry and the Edman degradation reaction. The Edman degradation is a very important reaction for protein sequencing because it allows the ordered amino acid composition of a protein to be discovered. It is also possible to generate an amino acid sequence from the DNA or mRNA sequence encoding the protein if this is known. In the first step, a portion of the protein is sequenced until a sequence of at least 6 contiguous amino acid is found for which the coding sequence is reasonably non-redundant. Such portion would contain amino acids encoded by relatively few different codons. All possible coding sequences of this region are then synthesized in a single batch. A nucleic acid sequence of at least 17 nucleotides is desired for specificity reasons. Alternatively, one either looks at codon use by the organism and generates an oligo or puts inosine at position of uncertainty. This pool of sequences is labeled and used in a southern analysis of the entire cell's restriction enzyme treated chromosome. Typically, one or a small number of restriction fragments will show hybridization indicating similarity to one particular probe in the pool. Having shown that there is a hybridizing sequence in the chromosome, the probe pool is then used to screen a random library of cloned chromosomal fragments. Alternatively, a new library can be generated using the chromosomal fragments of the same size as the hybridizing band. This enriched library would then be screened with the probe pool. Any clone that shows homology to the probe would likely carry at least a portion of the gene of interest. At this point, there are several possibilities. The first one is the region may be sequenced in order to determine the open reading frames or ORFs. Since you already know one end of the protein sequence, this will allow the conclusive identification of the desired gene. This sequence information can be used to clone a selectable marker, for example drug resistance, into the coding region for use in mutationally altering the chromosomal version. The second one is, alternatively, a restriction map of the cloned region can be produced that will indicate the relative position of the probe hybridizing region within the clone. The clone can now be mutagenized with transposons and such mutations analyzed physically to choose a pair flanking the probe hybridizing region. One of these must therefore be in the gene of interest. Isolation of gene or DNA fragment from cDNA libraries or complementary libraries. cDNA is a copy of double-stranded DNA complementary to messenger RNA or mRNA of the cell. This approach is used when we have information about expression of the gene. For gene isolation using this approach, usually a cDNA library is prepared. cDNA library is a collection of recombinant clones representing total mRNA of a cell or tissue at particular stage of development. The advantage of cDNA library is that it contains only the coding region of a genome. It is the method of choice for cloning eukaryotic genes in bacteria as they lack splicing mechanism. Preparation of cDNA library. The various steps for preparation of cDNA library are as follows. Isolation of mRNA. The total mRNA is isolated from the cell or tissue of interest where the desired gene is expressed. Because eukaryotic mRNAs contain a poly A tail at the 3' end, they can easily be separated from other types of RNA like rRNA and tRNA molecules present in the cell. Preparation of single-stranded cDNA. The enzyme reverse transcriptase is used to synthesize a DNA strand complementary to each mRNA molecule. A cDNA RNA hybrid is formed from which the RNA is removed with the help of RNase H or alkali. Preparation of double-stranded cDNA. The single-stranded cDNA has the property to form a transient hairpin loop at 3' end, which is used to prime the synthesis of second strand of cDNA by DNA polymerase I. After the single-stranded DNA molecules are converted into double-stranded DNA, the hairpin loop is removed with the help of S1 nuclease that acts only on single strands. Modification of ends. Ends of double-stranded cDNA are suitably modified by homopolymer tailing or addition of linkers and adapters to allow its insertion into a vector. Transformation of the host cells. The recombinant vectors containing cDNAs corresponding to mRNAs are introduced into suitable host cells. This collection of transformed host cells represents the cDNA library from that particular cell or tissue. The cDNA library is then screened for identification of the clone carrying the desired gene. The number of clones to be screened is given by the following formula. N is equal to ln into 1 minus p divided by ln into 1 minus 1 by n, where n represents the number of clones to be screened. P is the probability of finding a clone, ln represents natural log, and n is the number of mRNA molecules in the cell. The above described method for cDNA synthesis results in synthesis of shorter length of cDNAs due to removal of hairpin loop or incomplete copying of mRNAs. So, strategies are used to obtain full length cDNA molecules. One such strategy is to use a specially designed Escherichia coli vector to which mRNA is attached before copying and subsequent steps are followed while it is attached to vector. Similarly, use of S1 nuclease is avoided by adding poly G or poly C or poly T tail to 3' end of single-stranded cDNA. An oligonucleotide having sequence complementary to the tail is then used as a primer to synthesize double-stranded cDNA. A number of strategies are used for cloning cDNA depending upon information about expression of genes, that means whether mRNA is present in abundance or as low copy number or is a rare species. If purified mRNA is available for a gene, cDNA is directly prepared. However, for isolation of 29 cDNA clones in the moderate and low abundance class, it is usually necessary to construct a cDNA library. Some commonly used vectors for cDNA cloning are lambda insertion vectors, namely lambda GT10, lambda GT11 and ZAP2. cDNA libraries are tissue specific and are prepared using mRNA isolated from tissues where its gene is expressed. For example, for isolation of gene for insulin which is synthesized in pancreas, mRNA for construction of cDNA library is isolated from pancreatic cells and not from liver or other cells. Similarly, for isolation of genes for gliadin, a seed protein in wheat, cDNA library from developing wheat grains is used. Isolation of exons from cloned DNA by exon trapping. Exon trap is a novel method for the identification of signal sequences from genomic DNA. Exon trapping is an RNA polymerase chain reaction or PCR method to clone express sequences or exons directly from mammalian genomic DNA. Compared with other techniques for gene identification, exon trapping has two main characteristic features. First, it is independent of the availability of an RNA sample in which the gene to identify is expressed. Second, the sequences isolated directly derive from the input DNA. Some 10 to 20% of all genes might be expressed at very low levels or only during very short stages of development, making it difficult to isolate them based on their expression using cDNA hybridization or cDNA selection protocols. Exon trapping uses an assay isolating sequences based on the presence of functional splice sites. Consequently, sequences are isolated directly from the clone under analysis without knowledge or availability of tissues expressing the gene to be identified. Furthermore, because isolation is not based on hybridization, it is not possible to isolate highly similar sequences that derive from other parts of the genome not under analysis. The schematic representation of exon trapping is shown. Exon trapping starts with a vector that contains a strong promoter and has a single intron between two exons. When this vector is transfected into cells, its transcription generates large amounts of an RNA containing the sequences of the two exons. A restriction cloning site lies within the intron and is used to insert genomic fragments from a region of interest. If a fragment does not contain an exon, there is no change in the splicing pattern and the RNA contains only the same sequences as the parental vector. But if the genomic fragment contains an exon flanked by two partial intron sequences, the splicing sites on either side of this exon are recognized and the sequence of the exon is inserted into the RNA between the two exons of the vector. This can be detected readily by reverse transcribing the cytoplasmic RNA into cDNA and using PCR to amplify the sequences between the two exons of the vector. So, the appearance in the amplified population of sequences from the genomic fragment indicates that an exon has been trapped. Because introns are usually large and exons are small in animal cells, there is a high probability that a random piece of genomic DNA will contain the required structure of an exon surrounded by partial introns. Let us summarize the following discussions on gene isolation, isolation of genes from genomic DNA by cDNA and exon traps. Genes are the basic unit of a cell that passes on the traits of parents to the next generation. Genes are pieces of DNA or genes are part of a DNA molecule which codes for one polypeptide. A full understanding of a gene or the entire set of genes in a genome requires the isolation of genes. Once a gene is isolated, one can determine both its biochemical structures and its functions in an organism. Isolating the genetic material involves rupturing of cell membrane to release the cellular components and DNA, separation of the nucleic acid from other cellular components and purification of DNA. Isolation of a gene of interest involves cDNA libraries and exon trapping. Exon trap is a novel method for the identification of signal sequences from genomic DNA. Before we attend the next session, please try to answer the following questions. Write an overview on cDNA synthesis. Discuss the methods for isolation and purification of RNA. Write short note on purification of DNA. How to isolate a gene or DNA fragment of interest. Discuss the isolation of exons from cloned DNA by exon trapping. Here are some references. Introduction to Bioinformatics, Atwood and Parry Smith, Pearson Education. Bioinformatics, a beginner's guide by Jean Michael Clavier, John Wiley and Sons. Structural Bioinformatics by Philip E. Bioinformatics, Methods and Applications, Rastogi, SC and Rastogi P. Prentice, Hall of India Private Limited, New Delhi. Hope you have enjoyed the session. See you next time. Until then, goodbye.